Let us discuss about stem cells and niches in Drosophila germarium. This is a cross-section of Drosophila germarium. This cross-section of Drosophila germarium shows female germline stem cells, some somatic stem cells in their niches, and progeny cells derived from somatic stem cells. The germline stem cells produce cytoblasts. These cytoblasts undergo differentiation to produce oocytes. The somatic stem cells produce follicle cells. These follicle cells will make the egg cell. The cap cells create and maintain the niche for germline stem cells. The inner sheath cells produce the niche for somatic stem cells. In order to create germline stem cell niche, signaling event occurs between cap cells and germline stem cells. Now let us discuss about signals that create germline stem cell niche. The cap cells in Drosophila germarium produce TGF-beta proteins or transforming growth factor beta proteins DPP and GBB. The cap cells also produce HH protein. DPP and GBB proteins bind to TGF-beta receptor subunit 1 and TGF-beta receptor subunit 2 on the surface of germline stem cell. The HH protein in cap cell binds to the patch receptor or PTC on the surface of germline stem cell. Due to the binding of DPP, GBB and HH proteins to their receptors, transcription factors like MAD and MED cause expression of BAM gene to be repressed. Means the expression of BAM gene is upped by MAD and MED proteins. Due to the repression of BAM gene, germline stem cells undergo renewal. If the BAM gene is activated or the BAM gene is on, then the germline stem cells undergo differentiation. ARM and ZPG are two surface proteins. ARM and ZPG proteins physically link cap cells and germline stem cells. ARM and ZPG proteins are also important in maintaining the stem cell niche. Cells out of reach of ARM and ZPG proteins differentiate rather than renew. MicroRNAs are increasingly recognized as important regulators of cell differentiation, including germline cells. Some of the microRNAs bind to PUE protein present in the nucleus of cap cell. PUE protein is an essential germline regulator in both cap cells and germline stem cells. In order to create somatic stem cell niche, signaling event occurs between inner sheath cells and somatic stem cells. Now let us discuss about signals that create somatic stem cell niche. Wingless or WG protein is a WNT signal. Inner sheath cells produce WG signal. The plasma membrane of somatic stem cell has frizzled receptor or FZ. The WG signal produced by inner sheath cells is received by the frizzled receptor or FZ. Inner sheath cells produce HH signal. This HH signal is received by the patch receptor or PTC on the surface of somatic stem cell. Both frizzled receptor or FZ and patch receptor or PTC signal to control transcription. This transcription results in self-renewal of somatic stem cell. Both inner sheath cell and somatic stem cell produce ARM protein. The ARM protein of inner sheath cell interacts with ARM protein of somatic stem cell. Now, some important points. The germline stem cells produce oocytes and sperms. Stem cell niche is a location which contains a set of cells producing the signals. Please like, subscribe and share.